Imagine you're sitting in a hospital bed, right? And you need a blood transfusion.
Yeah.
The nurse comes in, hangs a bag of this deep red fluid, hooks it up to your AV and, opens the line. It looks incredibly simple. I mean, just fluid moving from a plastic bag right into your vein. But underneath that simple visual, there is just a microscopic universe of hidden codes.
Oh, yeah. Invisible clouds of electricity, shape-shifting antibodies.
Exactly. And right now, if a single biochemical mismatch happens, there is a literal microscopic war waiting to happen in your veins. We all think we know our blood. You sitting there listening, you probably know your type. Maybe you're A positive or O negative.
Right.
But that label it's barely scratching the surface.
It is the absolute definition of a biological iceberg. We like things to be neatly categorized, right?
Sure.
But the diagnostic landscape of immunohematology is incredibly murky. It's this space where genetics, physics, and immunology all just sort of collide. Because when we mix blood from two different human beings, we're initiating a really complex, high-stakes biological negotiation.
A negotiation, I like that.
Yeah. The science of compatibility isn't just about matching letters on a chart. It's about anticipating how millions of years of evolutionary defense mechanisms are going to react to foreign tissue. Because that's what a blood transfusion actually is. It's a liquid organ transplant.
Well, liquid organ transplant. That really puts it in perspective. And, today's mission on this deep dive is to decode that negotiation. We've got a whole stack of advanced immunohematology notes here, and we're going to break down the complex serological tests they do in a blood bank.
We really are.
We'll look at what these tests reveal about our genetics, map out all the different antibodies and the trends they show, and basically explore what actually happens inside the body when blood types clash. We're going far beyond just A, B, and O today.
Far beyond it.
So, okay, let's unpack this. To really grasp this, we have to start at the foundational blueprint of blood. If I go to donate blood and they tell me my type is, say, group A, I'm assuming that's just my phenotype, right?
Yes, exactly.
Like that's what the lab sees reacting in the test tube, but it's not my whole genetic story.
Right. Your phenotype is just the physical expression of your genetics. It's what the blood bank technologist actually observes when they mix your red blood cells with highly specific testing regions.
Okay.
So if your cells clump up a process we call agglutination when they're exposed to an anti-A reagent, well, your phenotype is group A.
Makes sense.
But your genotype, the actual genetic instructions you inherited from your parents, is hiding underneath that physical expression.
Yeah.
The inheritance of most blood group genes is codominant.
Codominant, right? So if you inherit the A gene from one parent and the B gene from the other, your cells just build both structures.
Exactly.
And your phenotype is AB. But group A is where it gets kind of interesting because the O gene is essentially, it's an amorph, it's a silent partner, right?
It is entirely silent, yeah. The O gene doesn't produce a functional enzyme to build an A or B antigen. So you could have an AA genotype, meaning you're homozygous for the A antigen.
Right.
Or you could have an AO genotype. In that case, only the A gene is physically expressing itself on the cell surface. The blood bank tests you, sees the A antigens, and types you as group A.
But genetically, you're secretly carrying that silent O.
Exactly. Which you can absolutely pass on to your
Yeah,
children.
which perfectly explains those, wild scenarios where two parents who have A and B blood respectively can somehow have a kid with group O blood.
Yep, it happens all the time.
Because if the parents are actually AO and BO genotypes, the child has a 25% chance of inheriting the silent O from both of them?
Exactly right.
But even within that group A phenotype, things aren't uniform. I was looking through the source material, and it splits group A into subgroups. So not all group A blood is created equal.
It certainly isn't. The group A phenotype is predominantly split into two subgroups. We call them A1 and A2. Roughly 80% of group A individuals are A1.
Okay.
And the remaining 20% are A2. And the difference isn't just some, trivial structural quirk. It's really about the density and the complexity of the carbohydrate chains on the surface of the red blood cell.
So what does that actually look like at the cellular level? Are the A2 antigens just physically smaller?
Well, it's more about efficiency. The enzyme that the A gene produces is called a transferase. Its whole job is to attach a specific sugar to the cell membrane.
Okay.
In an A1 individual, this transferase enzyme is highly efficient. It packs the red blood cell with up to 1 million A antigens, creating these highly branched, complex structures. They possess both a core A antigen and the highly complex A1 antigen.
Wow, 1 million.
Yeah. But in an A2 individual, the transferase enzyme has a slight mutation that makes it Much less efficient. They only manage to attach about a quarter of the antigens, and the structures remain scalar, more linear chains. They only possess the base A antigen.
But wait, if they both have the base A antigen, why does this matter for a transfusion? If I'm A2 and you give me A1 blood, my body still recognizes it as group A, right?
Not always.
Really?
Yeah, this is where the immune system's absolute intolerance for unfamiliar structures comes in. Because an A2 individual lacks that highly branched, complex A1 structure. Their immune system might look at a transfused A1 red blood cell and say, I recognize the base, but I have never seen this elaborate branching structure on top. This is foreign.
Oh, wow.
Consequently, a certain percentage of A2 individuals will actively form an anti-A1 antibody.
So a person with group A blood could literally reject blood from another person with group A blood.
They absolutely could.
This seems like a logistical nightmare for a blood bank. How do you even rapidly test for that difference if both of them will react to a standard anti-A region?
We use a remarkably elegant tool derived from nature, actually, a plant lectin. Yeah. Specifically, an extract from the seeds of a plant called Dolichos biflorus. Lectins are basically proteins that bind to very specific carbohydrate structures.
Wait. We're using seed extracts to cross-match human blood.
We are. The ACOS's specificity comes from a very specific immunodominant sugar called N-acetyl D-galactosamine.
That is quite the name.
It's a mouthful. But the Dolichos biflorus lectin has an extraordinary affinity for the dense, highly branched N-acetyl D-galactosamine structures that are found on A1 cells.
Oh, I see.
So when we mix this lectin with A1 blood, it agglutinates strongly, it clumps up. But when we mix it with A2 blood, the antigens are too sparse and too simple. There's no reaction.
That's brilliant.
It really is. It allows us to instantly differentiate the subgroups and prevent a highly specific transfusion reaction.
It's incredible that we rely on a plant to detect a microscopic sugar variation on a human cell. But, okay, let's dig deeper into that structural foundation. You mentioned the base A antigen. If we look at the genetics, everyone, AB, and O, they all actually start with the exact same cellular foundation, right? The H antigen.
Yes, exactly. You kind of have to picture blood group antigens as an assembly line. Before you can attach the A sugar or the B sugar, you need a docking port.
Okay.
And that docking port is the H antigen. Genetically, almost all humans inherit the FUT1 gene. That gene builds this H antigen by attaching a sugar called fucose to the red blood cell membrane.
So instead of a concrete slab, the H antigen is kind of like the universal docking port on a space station.
I love that analogy.
Yeah, so if you have the A gene, your enzymes attach the A module, that long N-acetyl whatever, onto the docking port. If you have the B gene, your enzymes attach a different module, D-galactose, onto that exact same port.
That is a perfect way to visualize it. And if you have the O gene, which, remember, produces a non-functional enzyme, you don't attach any modules at all.
You just leave the universal docking port empty and exposed.
Exactly.
Which leads to a really counterintuitive fact, I think. Group O blood, which we usually think of as having nothing on it, actually has the most exposed H antigen of any blood type.
Precisely. Because there are no A or B modules crowding the surface, the H antigens are entirely accessible.
Right.
Conversely, a person who is group A1B has the least amount of exposed H antigen.
Because their highly efficient enzymes have basically attached A and B modules to almost every single available docking port.
Exactly.
Okay, so everyone has this universal docking port, but there is a massive exception that completely breaks this rule, right? The Bombay phenotype.
Oh, yes.
If the H antigen is the universal docking port, what is happening with a Bombay individual? Is it just an empty lot?
A Bombay individual is missing the docking port entirely. It is one of the absolute rarest blood phenotypes in the world. They inherit 2 recessive H genes, so their genotype is little h, little h. They have a mutation in that FUT1 gene we talked about, meaning they completely lack the ability to attach that foundational fucose sugar to the cell membrane.
So even if they inherited the A or B genes from their parents, those transferase enzymes are just floating around with absolutely nothing to attach the sugars to.
Exactly. The enzymes are there, they're ready to work, but the assembly line is broken at the very first step.
Man.
Because they can't attach the A or B sugars without the H antigen docking port. Their red blood cells have 0 A, B, or H antigens on them.
But wait, if they have no A or B antigens, a standard blood bank test is going to see them as group O.
And that is exactly what happens initially. Routine anti-A and anti-B regions will show no reaction, so the blood type is O. But giving a Bombay individual standard group O blood is a catastrophic, usually fatal mistake.
Because group O blood is absolutely covered in those exposed H antigen docking cords. And since the Bombay individual has never had an H antigen in their entire life, their immune system sees it as a massive hostile invasion.
Right. Their immune system naturally produces anti-A, anti-B, and crucially, an incredibly potent anti-H antibody.
Oh, no.
The moment you infuse normal group O blood, their anti-H antibodies will violently attack and destroy those donor cells right inside the blood vessels. The only compatible blood for a Bombay patient is from another Bombay individual.
But if their immune system attacks O blood and they're, say, in a car crash, what do you even do? I mean, you can't just synthesize Bombay blood in an emergency room.
You can't. It is a profound logistical crisis. If a Bombay individual requires a transfusion, blood banks have to activate global rare donor registries.
Global? Wow.
Yeah, we often have to fly frozen units of Bombay blood across international borders. It's why people with this phenotype are strongly encouraged to donate and freeze their own blood in autologous programs just in case they ever need surgery.
That perfectly illustrates why relying just on a surface-level phenotype can be a fatal trap. And I think that brings us to the other major identifier on our red blood cells, which is the Rh factor. We all hear about being A positive or O negative, but positive and negative is a massive oversimplification, isn't it?
It is a severe reduction of a highly complex genetic system. When the general public says they are Rh positive, they're referring to the presence of just one specific protein, the D antigen.
Okay.
But the Rh blood group system actually consists of over 50 different antigens. In routine practice, we focus on the big five: D, big C, little c, big E, and little e.
The history behind how we name these is wild, by the way. There was a massive scientific debate, right? The Wiener nomenclature versus the Fisher race theory.
It was one of the great debates in early immunohematology. In the 1940s, Ronald Fisher and Robert Race proposed a genetic theory that the Rh antigens were controlled by 3 closely linked genes on a chromosome. They thought it was inherited as a package deal: D or D, C or C, and E or E.
It's clean, it's logical.
It is. And it's how we primarily teach it today.
But Alexander Wiener had a completely different idea.
He did. Wiener believed that the Rh system was controlled by a single massive gene locus that produced one single complex antigen and a glutinogen that just happened to have multiple different structural factors protruding from it.
So instead of individual letters, he used a system of Rs, Rhs, and prime symbols, right?
For instance, what Fisher race called little c, Wiener called HR prime, what we call the D antigen, Wiener called RH0.
So who is actually right?
Interestingly, modern molecular genetics proved they were both a little bit right and a little bit wrong.
Of course.
We now know there are 2 closely linked genes: the RHD gene, which codes for the D antigen, and the RHCE gene, which codes for the C and E combinations. So it isn't 3 genes like Fisher race thought, but it isn't one single gene like Wiener thought either.
But we still use both systems.
We do. Because Wiener's notation was heavily published in the early days, blood bank technologists today still have to be fluent in both languages.
So if a tech receives a unit of blood from a rare donor registry, and the paperwork says the genotype is RR in Wiener notation, they need to instantly translate that.
Yes. They need to know that RR translates to little d, little e, little c, little e over little d, little c, little e in Fisher race.
No.
Meaning this donor's red blood cells completely lack the D antigen, the big C antigen, and the big E antigen.
Why is that specific RR combination so critical?
Because it is the ultimate stealth blood for patients with complex immune profiles. Yeah. About 14% of the white donor population has this RR genotype.
Okay.
If a patient requires multiple transfusions over their lifetime, say a patient with sickle cell disease, they might develop a low antibody.
And the low antibodies are immune weapons formed against foreign blood.
Exactly. And the 3 most highly immunogenic targets in the Rh system are D, big C, and big E. If a patient has formed antibodies against all 3 of those, this R blood is the only safe option because it lacks all of those targets. It flies completely under the radar.
Wow. Let's focus on the D antigen for a second, because that's what determines if you are Rh positive or negative. Is it really a binary switch? You either have it or you don't.
No, and this is where it gets highly technical. The D antigen is an enormous complex protein that weaves in and out of the red blood cell membrane. Imagine it as a massive mosaic made up of dozens of individual structural epitopes or puzzle pieces.
Here's where it gets really interesting. What happens if someone is missing a few pieces of the puzzle?
We call that a partial D, or a D mosaic.
Yeah.
Genetically, they possess the D antigen, but it is incomplete.
Okay.
If we test their blood with a standard, highly potent anti-D reagent, it might still grab onto the pieces that are there, and the test will show up as weakly positive. Historically, we called this a weak D.
But here is the danger, right? If they're Rh positive, you would normally give them Rh positive blood. But if they're a partial D and you give them complete Rh positive blood, their immune system is going to look at the puzzle pieces they are missing.
Exactly. Their immune system has never been exposed to those specific missing epitopes. When they receive a transfusion of normal, complete Rh positive blood, their immune system recognizes those missing puzzle pieces as a foreign invasion. Wow. They will actively mount an immune response and form an anti-D antibody against those specific parts of the D antigen, even though they themselves type as Rh positive. It is a stunning display of the immune system's precision.
That is just wild. But wait, if they only react weakly to the lab tests, how do we confidently catch a weak D? Because I know the testing methodology for Rh is totally different than for ABO.
It comes down to the nature of the antibodies. ABO antibodies are naturally occurring. You develop them early in life just by being exposed to environmental bacteria that have similar carbohydrate structures.
Right, there's there.
Yeah. Because they're naturally occurring, they are large, robust, and react at room temperature. You just mix the blood and the reagent on a slide, and it clumps visibly. But Rh antibodies are immune antibodies.
Meaning you only produce them if you are specifically exposed to foreign human blood.
Exactly.
Yeah.
Either through a transfusion or during pregnancy.
And because they are immune antibodies, they are physically different.
Yes. To force these immune antibodies to react in a laboratory setting, we have to mimic the conditions of the human body. We incubate the sample at 37 degrees Celsius.
Or body temperature.
Right. And to detect a weak D, we have to perform a highly sensitive procedure called the indirect antiglobulin test, or IAT.
But I understand that modern automated lab equipment can actually miss this. Like, how does a $1 million automated gel system completely miss a weak D?
It's a fascinating limitation of the technology. The traditional IAT procedure requires a crucial step, the wash phase.
The wash phase.
You incubate the patient's cells with the reagent antibody, giving them time to bind. But before you can add the final indicator reagent to see if binding occurred, you absolutely must wash the cells with saline multiple times.
Why? What are you washing away?
You are washing away all the unbound, free-floating antibodies in the surrounding plasma. If you don't wash them away, when you add your final indicator, anti-human globulin, or AHG, that indicator will bind to the free-floating proteins instead of the ones attached to the red blood cells. It neutralizes your reagent, causing a false negative.
So the indicator gets distracted by the garbage in the plasma and totally misses the actual target on the cell.
Exactly. Now, automated gel systems are brilliant. They use a microtubule filled with gel that already has the AHG embedded in it. As the centrifuge spins, the red blood cells get pushed through the gel. The free-floating proteins stay at the top, effectively separating them without a manual wash phase.
Okay, that sounds better. What's the catch?
Here is the catch. Many automated systems are programmed to run routine ABO and Rh typing as a rapid direct test without the 37-degree incubation and without pushing it through an AHG gel matrix, just to save time.
Oh, wow. So if the system skips the incubation and the AHG phase for routine screening, the weak D never gets forced to react. The machine just spits out Rh negative.
Precisely. And if that person is a blood donor, their unit gets labeled as Rh negative. If that unit is transfused into a truly Rh negative patient, that patient's immune system will detect those weak D puzzle pieces and mount a severe immune response.
It's a perfect example of why technology can never fully replace a technologist's actual understanding of the underlying biochemistry.
I couldn't agree more.
Which is a perfect segue. We've talked extensively about the antigens the targets sitting on the red blood cells. Now we need to look at the weapons the body uses to attack those targets. Welcome to the antibody zoo.
The antibody zoo. It's an apt name because the plasma is filled with different families of antibodies, and they all have distinct behaviors, preferences, and environmental requirements. In the blood bank, we are primarily dealing with two classes of immunoglobulins, IgM and IgG.
Let's break down the physical architecture of these two because the difference is staggering.
Structurally, an IgG molecule, the typical immune antibody, is a monomer. It looks like a single Y shape with two tiny binding sites at the tips of the Y.
So it's small.
It is a relatively small, compact protein. But an IgM molecule, the naturally occurring ABO antibody, is a pentamer. It is literally 5 of those Y shapes joined together at the base, forming a massive snowflake-like ring with 10 binding sites pointing outward in all directions.
So if we're trying to visualize this, IgM is like a giant microscopic grappling hook capable of snagging 10 different red blood cells at once in the cold.
Yes.
While IgG is just a tiny pair of tweezers that needs the cells to be warmed up and shoved together to grab anything.
That is exactly how they function mechanically. Because IgM is so massive and has a span of about 35 nanometers, it can easily reach across the plasma and grab multiple red blood cells simultaneously. It creates a physical web, causing direct visible clumping at room temperature.
But the tiny IgG tweezers...
They have a wingspan of only about 14 nanometers, and that size limitation introduces a massive problem for us because of a concept called the zeta potential.
Right, zeta potential.
Yeah.
You were telling me our red blood cells are actively trying to repel each other.
They are. The membrane of a red blood cell is studded with molecules called sialic acid, which carry a net negative electrical charge.
Okay.
When red blood cells are suspended in plasma, that negative charge attracts a dense cloud of positively charged ions from the surrounding fluid. This forms an electrical double layer, often referred to as the Gouy-Chapman layer.
So every single red blood cell is flying around surrounded by a microscopic electrical force field.
Exactly. This force field, the zeta potential, ensures that the red blood cells maintain a physical distance of about 25 nanometers from each other. They literally bounce off one another.
Because if they didn't, the billions of cells in your veins would just clump together into a solid lethal block.
That's right.
Okay, let's do the math here. If the force field keeps the cells 25 nanometers apart, but the IgG tweezers are only 14 nanometers wide, the antibody is too short.
Yes.
It can latch onto one cell, but it physically cannot reach across the void to grab a second cell.
You've hit on the fundamental challenge of modern blood banking. An IgG antibody, like a dangerous Rh antibody, can heavily coat a patient's red blood cells. But because of the zeta potential, we won't see any clumping in the test tube.
It'll just look fine.
The blood will look perfectly compatible. To overcome this physics problem, we have to manipulate the chemical environment using enhancement mediums. The most common one is LISS, or low ionic strength solution.
So how does LISS actually hack the force field?
Well, normal saline has a sodium chloride concentration of 0.9%. LISS drops that concentration dramatically to 0.2%. By stripping the environment of those free-floating ions, LISS physically shrinks the size of the ionic clouds surrounding the red blood cell.
It shrinks the force field.
Right. It lowers the dielectric constant of the solution, allowing the cells to move much closer together. We also add substances like PEG, polyethylene glycol, which absorbs water molecules and physically forces the antibodies in red cells into a tighter space.
So we are literally dehydrating the environment and altering electrical charges just to give these tiny tweezers a chance to grab hold. That is wild.
It is chemistry at its finest.
Now, what about the dosage effect? This is another quirk of how antibodies behave, and it ties right back into genetics.
It does. Think back to codominant inheritance. Let's look at the M&S blood group system. If you inherit the M gene from both parents, your genotype is MM. You are homozygous.
Okay.
Because you only have the instructions to build M, your red blood cells will manufacture roughly 1 million M antigens on their surface. You have a massive concentrated dose of the target.
But if I inherit M from my mom and N from my dad?
Then you are heterozygous, MN. Your cellular machinery has to split its resources. It builds 500,000 M antigens and 500,000 N antigens.
So the dosage of the M target is cut completely in half.
Exactly. And certain antibodies in RZU, specifically those in the MNS, Duffy, KID, and RH systems, are highly sensitive to this receptor density. If a technologist is testing a patient's plasma that contains a weak anti-M antibody against a homozygous MM cell, the antibody sees a massive herd of targets. It binds aggressively, giving a strong, unmistakable 4+ positive reaction.
But if they test that exact same plasma against a heterozygous MN cell...
The antibody looks at the sparse, spread-out targets and barely reacts. It might give a weak microscopic reaction, or it might be completely negative.
Wow.
This is a terrifying phenomenon when you are trying to identify an unknown antibody, because the antibody appears to vanish and reappear depending on the genetic makeup of the random test cells you were using.
It's a predator that only attacks if the herd is tightly packed.
Exactly.
Okay, let's go on a tour of specific enclosures in this antibody zoo, because some of these behaviors are borderline science fiction. Let's start with the Lewis antibodies, anti-LEA and anti-LEB.
The Lewis system is entirely unique because Lewis antigens are not actually built on the red blood cell.
Wait, what?
They're manufactured by tissue cells and secreted directly into the plasma and other body fluids. As red blood cells circulate through the plasma, these floating Lewis carbohydrate chains just sort of passively absorb or stick onto the red blood cell membrane.
They're hitchhikers.
Exactly, they're hitchhikers. And because they're just loosely stuck to the membrane, and the antibodies they provoke are usually cold-reacting IgM grappling hooks, they're rarely a danger during a transfusion. The transfused cells often just shed the antigens anyway.
But I'm guessing they cause problems in the lab.
Oh, they're incredibly annoying in the lab. A bulky IgM Lewis antibody will cause massive clumping and mask the presence of a truly dangerous IgG antibody hiding underneath.
But we have a biological hack for this, right? Neutralization via saliva.
It sounds like an archaic apothecary trick, but it's pure biochemistry. Because Lewis antigens are secreted into all body fluids, the saliva of a person who possesses the Lewis gene is packed with free-floating Lewis antigens. If we take that saliva and mix it with our patient's plasma, the Lewis antibodies in the plasma will immediately latch onto the antigens in the saliva.
They get completely bogged down fighting the saliva.
Yes, the antibody is neutralized. We can then take that neutralized plasma, test it against red blood cells, and clearly see if there's an Rh or a KIT antibody hiding in the background because the Lewis antibody has been completely blinded.
That is brilliant. You just throw chaff into the air to confuse the radar. Okay, what about anti-P1? The source material says it can be neutralized by hydated cyst fluid. What exactly is a hydated cyst?
This is where immunohematology meets parasitology. The P1 antigen is a carbohydrate chain. Nature tends to reuse successful structures, and it turns out that the Echinococcus granulosus tapeworm.
The tapeworm, oh boy.
Yeah. The tapeworm, which infects humans and forms massive fluid-filled hydated cysts in the liver and lungs, produces a glycoprotein that is structurally identical to the human P1 antigen.
So, just like the saliva trick, if a lab is struggling with an annoying anti-P1 antibody, they can literally acquire fluid extracted from a tapeworm cyst, drop it into the human plasma, and neutralize the antibody.
Precisely. The antibody binds to the parasitic substance, leaving the plasma clear for further testing.
That is unsettling, but amazing.
Let's move to anti-I. The source notes it as a cold autoantibody that reacts with adult cells, but not with cord blood cells.
The I system is a beautiful illustration of human cellular development. When a fetus is developing in the womb, the precursor chains on their red blood cells are straight, unbranched, linear structures.
Okay.
We call this the little I antigen. Newborn cord blood is covered in little I. But over the first 18 months of life, as the child's enzymes mature, those linear chains branch out, creating a complex, highly branched structure. This is the big I antigen. Almost all adults possess big I.
So if an adult develops an autoimmune condition, say following a mycoplasma pneumonia infection, and their body starts making an antibody against big I, their blood will just clump up as soon as it cools down in the lab. Yes, it's a cold autoantibody. And it is a nightmare for blood banks because it will agglutinate essentially every adult donor unit in inventory, since all adults have big I.
Right. So how do you prove what it is?
To prove that the culprit is specifically anti-I, we test the adult patient's plasma against cord blood cells from a newborn.
Ah, because the newborn cells only have the linear little I chains.
Exactly. The adult's anti-big I antibody will look at the cord cells, fail to recognize the simple linear structure, and ignore them. A negative reaction with cord blood confirms the antibody's identity.
Wow. Okay, we've covered the hitchhikers and the autoantibodies. Now we need to talk about the apex predators of the zoo. The KID antibodies, anti-JOCA and anti-JKD, these are notorious.
If you want to terrify a seasoned blood bank technologist, just whisper the word KID.
Really?
They are the absolute ninjas of the antibody world. The KID antigens are actually urea transport proteins on the red cell membrane. But the antibodies formed against them are infamous for causing severe delayed hemolytic transfusion reactions.
Why are they delayed? Like, why doesn't the reaction happen while the blood is hanging on the IV pool?
It is entirely due to a phenomenon called the anamnestic response, or immunological memory.
Okay.
Let's say you receive a blood transfusion containing the JKK antigen. Your immune system recognizes it as foreign and forms anti-JNCA. You have a mild primary immune response.
Right.
But KID antibodies are highly dependent on continuous antigen stimulation. Over a few months or years, as those donor cells die off, the level of anti-JNCA in your plasma plummets. It drops so low that it falls completely below the detection threshold of our laboratory instruments.
So 10 years later, I come into the hospital, you draw my blood, run a standard antibody screen, and the computer tells you my blood is perfectly clear.
Yep.
You have no idea the anti-JNK is there.
None. The screen is negative. So we cross-match a unit of blood that happens to possess the JK antigen.
Yeah.
It looks perfectly compatible in the test tube. We transfuse it, you feel fine, and you go home.
Yeah.
But inside your lymph nodes, your memory B cells, the historians of your immune system, recognize the JK antigen immediately.
The alarm bells ring.
They don't just ring. It is a full-scale mobilization. Within 3 to 7 days, those memory cells rapidly differentiate into plasma cells and flood your bloodstream with massive amounts of fresh, highly destructive IgG anti-JK.
Wow.
This is the anamnestic response. Because it's a secondary response, it is exponentially faster and more violent than the first time. The new antibody army hunts down every single transfused red blood cell and flags them for destruction, causing a massive delayed hemolytic reaction days after you left the hospital.
That is genuinely terrifying. How do you defend against an enemy that actively hides from your tests?
Our primary defense is exhaustive record keeping. If a patient ever had a KID antibody identified, even a faint trace of it 20 years ago at a different hospital, it must be permanently logged in their medical record. Once a KID, always a KID.
So you just always avoid the antigen.
We must provide JK-negative blood for the rest of their life, regardless of what today's test says. In the lab, we can also use proteolytic enzymes, like physin or papain, which strip away neighboring glycoproteins on the red cell membrane, effectively exposing the KID antigens and making those sneaky, low-titer antibodies easier to detect.
One more exhibit in the zoo, the Doneth-Landsteiner antibody. It causes paroxysmal cold hemoglobinuria, or PCH, and it is a biphasic hemolysin. Let's unpack that.
This is perhaps the most mechanically complex antibody we encounter. Biphasic means it requires 2 completely distinct temperature phases to execute its attack. The Doneth-Landsteiner antibody is an autoantibody directed against the P antigen, which is present on almost all human red cells.
So it attacks your own blood, but only under specific weather conditions.
Quite literally. In patients with PCH, when they're exposed to cold temperatures, perhaps they go outside without gloves in the winter, the blood circulating in their extremities cools down to around 20 degrees Celsius. During this cold phase, the Doneth-Landsteiner IgG antibody binds to the P antigen on the red blood cells.
Yeah.
But it doesn't destroy them yet. It just attaches and sets a trap by binding the first component of the complement cascade, C1q.
It's just painting the target.
Exactly. Now, as that blood circulates out of the cold hands and travels back into the warm core of the body, the temperature rises back to 37 degrees Celsius.
The trap is sprung.
Yes. The antibody itself might actually detach in the warmth, but the warming triggers the rest of the complement cascade to activate sequentially. The proteins form a membrane attack complex, a literal microscopic pore that punches a hole straight through the red blood cell membrane.
So the cells violently explode right inside the veins.
Yes. Intravascular hemolysis. The red cells lyse, releasing massive amounts of free hemoglobin into the blood, which is eventually filtered by the kidneys, resulting in hemoglobinuria, dark, bloody urine. It binds in the cold and destroys in the warm.
Okay, let's take a breath and step back. We know all these stealthy antibodies exist. We have cold-reacting grappling hooks, biphasic traps, and KID ninjas that disappear from the plasma. With all these threats lurking, how do we actually prove a bag of donor blood won't trigger a lethal reaction before we hang the IV bag? That brings us to compatibility testing and the cross-match.
The ultimate final failsafe before a transfusion is the major cross-match. All the theory we just discussed has to translate into a physical demonstration of safety.
Right. So the major cross-match specifically tests the recipient's plasma against the donor's red blood cells. But wait, why aren't we testing the donor's plasma against the recipient's cells? The donor might have crazy antibodies too, right?
They absolutely might.
Right.
But we rely on the major cross-match because of the physical realities of modern medicine. Decades ago, we transfused whole bloods. The red cells and the plasma all mixed together. But today, a blood donation is centrifuged and separated into components. If you are anemic, you don't receive whole blood, you receive packed red blood cells. We have physically squeezed out and removed almost all of the donor's plasma.
So we have effectively removed the donor's weapons.
Exactly. Since the donor's antibodies are mostly gone, we aren't worried about the donor attacking the patient. However, the patient still has their entire volume of plasma fully loaded with their own antibody arsenal.
Yeah.
The primary threat is the recipient's immune system destroying the incoming donor red cells. Therefore, we must mix the recipient's plasma with the donor cells in a test tube to simulate what will happen in the vein. That is the major cross-match.
Which leads to the universal rules everyone learns. Group AB individuals are the universal recipients because they don't have naturally occurring anti-A or anti-B in their plasma to attack anything. And group O negative is the universal donor because those cells are stripped of A, B, and D antigens. There are no targets for the recipient to attack.
Those are the broad strokes, yes. In a trauma situation, when a patient is bleeding out and we don't have 10 minutes to figure out their type, we release uncross-matched Group O blood. O negative for females of childbearing age to protect future pregnancies, and often O positive for males to preserve the precious O negative inventory. But routine blood banking operates on a much more nuanced compatibility matrix.
Right. Let's look at a scenario. What if a patient types as AB positive, but the hospital's blood bank is completely out of AB positive blood and only has a negative blood on the shelf? Can you give a negative blood to an AB positive patient?
You can, and we do it every day. The fundamental governing logic of compatibility is this. You must avoid exposing a patient to an antigen they lack.
Okay, let's break that down.
Let's look at the patient. They're AB positive. This means they possess the A antigen, the B antigen, and the D antigen. They have all the major targets.
So if you give them A negative blood, you are just giving them the A antigen, which they already have, and you are withholding the D antigen. You aren't introducing anything foreign.
Exactly. Their immune system looks at the A-negative cells and sees familiar structures. Now, a sharp student might point out, wait, isn't there a tiny bit of residual plasma left in that bag of A-negative packed cells? And doesn't A-negative plasma contain anti-B, which could attack the patient's B antigens?
Yeah, you said almost all the plasma is removed, but not all of it?
That is the minor incompatibility. But the volume of residual plasma in a packed red cell unit is so small, maybe 20 to 30 milliliters, that when it enters the recipient's entire 5-liter circulatory volume, it is instantly diluted. The concentration of anti-B drops to harmless levels. It is a medically acceptable practice.
Okay, let's say you run a cross-match, and it fails. The patient's plasma aggressively clumps the donor cells in the tube. How do you figure out if the patient has an alloantibody, meaning they are attacking foreign blood, or if they have an autoantibody where they are attacking their own blood?
We run a critical control test called the direct antiglobulin test, or DAD. This is the autocontrol. Instead of mixing the patient's plasma with donor cells, we test the patient's red blood cells directly to see if they are already coated with their own antibodies in vivo inside their body.
If the D is positive, their own cells are coated. They have turned their weapons on themselves.
Correct. That's an autoimmune hemolytic anemia or a cold autoantibody like the anti-WAP we discussed earlier. But if the DAT is completely negative, meaning their own cells are clean, then the antibody destroying the donor cells in the cross-match must be an alone antibody. It is an immune response directed purely at foreign targets.
So you know it's an antibody. You run an antibody identification panel, and it looks like it's an anti-KID. How do you absolutely prove it before you sign your name on the blood release form? I mean, you can't just guess.
Blood banking is deeply rooted in statistics. We use the 3x3 rule. This rule dictates that to conclusively identify an antibody with a 95% confidence interval, we must observe exactly 3 positive reactions with distinct test cells that we know possess the target antigen.
You have to watch the antibody attack the target 3 separate times.
Yes, and equally importantly, we must observe exactly 3 completely negative reactions with test cells that we know lack the target antigen.
You have to watch the antibody walk past an empty target 3 times just to prove it isn't wildly attacking everything in sight.
Precisely. If it hits the target 3 times and ignores the empty space 3 times, mathematically, it is not a coincidence. Now, to actually see these reactions, especially for the tiny IgG antibodies, we use that indicator reagent we mentioned earlier, polyspecific anti-human globulin, or AHG.
We talked about how AHG acts as a bridge to overcome the zeta potential, but why is it called polyspecific?
Because it is a cocktail designed to catch 2 different pieces of evidence. It contains anti-IgG, which is an antibody directed against human antibodies. It physically grabs the FEC portion of 2 different IgG molecules that are attached to red cells, linking them together to create visible agglutination.
Okay.
But the polyspecific cocktail also contains anti-C3.
C3 is a piece of the complement cascade, right?
Yes. Sometimes a particularly aggressive antibody, like a KID, will bind to a red cell, immediately activate the complement cascade to destroy it, and then the antibody itself might detach and float away.
The antibody leaves the scene of the crime.
Exactly. But the complement cascade leaves a permanent scar on the red cell membrane. specifically a degraded protein fragment called C3rd. If our AHG region only contained anti-Ig, we would get a false negative because the antibody's gone.
But the anti-C3rd catches the scar.
Yes. Because polyspecific AHG includes anti-C3rd, it binds to that complement scar, proving unequivocally that an antibody interaction did occur.
It's like finding a bullet hole in the wall after the shooter has run away. The footprint remains. Now, in modern hospitals, a lot of this tube mixing isn't even happening. The concept of electronic cross-matching is widespread. Are we seriously just letting an algorithm decide if blood is safe?
We are relying on an algorithm, but the gates to access that algorithm are heavily fortified. Electronic cross-matching, where the computer simply verifies that the patient's ABO type is compatible with the donor's ABO type, is only legally permissible under incredibly strict criteria.
Like what?
First, the computer system itself must have validated, hard-coded logic that physically locks the user out if they attempt to issue incompatible blood.
But the computer is only as smart as the data entered into it if someone typos an A instead of an O when they log the patient's type.
Which brings us to the second non-negotiable rule. A patient must have 2 separate concordant ABO blood types on file before the computer is allowed to perform the cross-match. One type must be from the current blood sample drawn for this admission. The second type must be independently verified. It can be a historical record from a previous hospitalization, or, if they are a new patient, a nurse must draw a completely separate second tube of blood at a different time.
So 2 completely different humans have to draw the blood, test the blood, and agree on the blood type before the algorithm is unlocked.
Yes. And furthermore, the patient's antibody screen must be entirely negative. If there is any histories, past or present, of an antibody, even a harmless Lewis antibody, the electronic cross-match is immediately aborted, and the technologist must perform a full manual AHG cross-match in the test tube.
The layers of safety, the redundancy, it's monumental. But despite the computers, the 3x3 rules, and the AHG bridges, things still slip through the cracks. The body reacts in ways we couldn't predict, or someone makes a fatal error. Let's talk about when it all goes wrong. Transfusion reactions.
It is the absolute nightmare scenario. And despite all the biochemical mastery we have just discussed, the most sobering statistic in immunohematology is the cause of these reactions.
Right. It's not the Doneth-Landsteiner trap. It's not the kid ninja. Over 90% of severe transfusion reactions are caused by clerical errors. A typo. Someone writes down the wrong name on a tube, a nurse distractedly scans the wrong wristband, or the blood bag gets handed to the patient in bed A instead of bed B.
Human error is, and always will be, the weakest link in the chain, which is why the universal protocol, the moment a patient spikes a fever or reports back pain during a transfusion, is to stop the infusion immediately, leave the IV line open with saline, and painstakingly recheck every single piece of paperwork.
You verify everything.
You verify the wristband, the blood bag tag, the laboratory printout. If there is a clerical mismatch, you assume an ABO incompatibility is actively occurring. But if the paperwork is perfect, we have to become physiological detectives.
Let's run through the different profiles of these reactions. Let's start with the most explosive allergic reaction, anaphylaxis.
Anaphylactic reactions are terrifying because they happen within seconds to minutes of the transfusion starting, often after only a few milliliters of blood have entered the vein. The patient doesn't spike a fever.
Okay, so no fever. What are the symptoms?
Instead, they experience profound hypotension, a crash in blood pressure, bronchospasm, and they physically cannot breathe.
Why does this happen? Is it an ABO mismatch?
No, it has nothing to do with red blood cell antigens. It almost exclusively occurs in a very specific population.
Yeah.
Individuals who are genetically deficient in IgA.
IgA is a different class of antibody. It's the mucosal one, right? Found in saliva and tears.
Yes, and it circulates in normal plasma. But a small percentage of the population lacks the genetic ability to produce IgA. Because they have never possessed it, if they were ever exposed to it, perhaps through a prior transfusion or during pregnancy, their immune system recognizes the IgA protein as a highly foreign invader and creates a potent anti-IgA antibody.
So if they receive a standard bag of packed red blood cells.
Even though we remove most of the plasma, the residual 20 milliliters of donor plasma is packed with normal donor IgA. The moment that plasma hits the patient's bloodstream, their anti-IgA antibodies cross-link with the donor IgA, triggering massive mast cell degranulation.
Wow.
A tidal wave of histamine is released, causing the anaphylactic shock.
So how do you ever give them blood?
We must mathematically eliminate the donor plasma. We take the donor red blood cells and put them into an automated cell washer. We wash the cells with liters of sterile saline, spinning and decanting repeatedly until every single molecule of donor IgA has been flushed away. Only then are the washed red blood cells safe to transfuse.
Wow. Okay, next up is the most common reaction, the febrile non-hemolytic reaction. The hallmark here is a sudden temperature spike of more than 1 degree Celsius.
Febrile indicates a fever, and non-hemolytic is the critical distinction. It means the red blood cells themselves are not being destroyed. This reaction is usually caused by the patient's immune system attacking something else in the bag, the donor's white blood cells, or leukocytes.
Wait, I thought packed red blood cells were just red cells.
We filter them to remove leukocytes, a process called leukoreduction, but it isn't 100% perfect. Some donor white cells survive. If a patient has received multiple transfusions in the past, they develop HLA antibodies against foreign white cells.
Okay.
When the donor white cells enter the bloodstream, the patient's antibodies attack them. As the white cells are destroyed, they rupture and release cytokines, chemical messengers. These cytokines travel directly to the patient's hypothalamus in the brain and trigger it to raise the core body temperature.
How do you prove it's non-hemolytic?
Yeah.
Because a fever is also the first sign that red cells are exploding in an ABO mismatch. You can't just assume it's the white cell.
Exactly. You must draw a post-transfusion blood sample and send it to the lab immediately. We spin the tube in a centrifuge to separate the plasma from the cells. If the red cells were exploding intravascularly, they would spill their red hemoglobin into the plasma, turning it bright pink or red. If the plasma is a clear straw yellow color, we can breathe a sigh of relief. Second, we run a direct antiglobulin test, the DT. If it is negative, there are no antibodies coating the circulating red cells. The fever is non-hemolytic.
Okay, let's look at the dark side of that coin. Hemolysis. Specifically, the delayed hemolytic reaction. This brings us right back to our kid ninjas.
It does. The scenario plays out exactly as we discussed with the anamnestic response. The patient receives the blood, they feel completely fine, and they are discharged. But 3 to 10 days later, they return to the emergency room. They have a mild fever, their hemoglobin levels have inexplicably crashed, and their skin and eyes have turned yellow. They are jaundiced.
Why the jaundice? Why doesn't their urine just turn bloody like the cold autoantibody trap?
Because of where the destruction is taking place. This is the difference between intravascular and extravascular hemolysis. The Donner-Flansteiner trap causes intravascular hemolysis. The cells are blown to pieces right inside the blood vessels by complement.
But delayed reactions.
Delayed reactions, driven by IgG antibodies like anti-JACA, primarily cause extravascular hemolysis.
Extravascular meaning outside the blood vessels. So where are the cells going to die?
They are being dragged into the reticuloendothelial system, primarily the spleen and the liver. The IgG antibodies coat the donor red blood cells. As these coating cells squeeze through the incredibly narrow sinusoidal capillaries of the spleen, the resident macrophages, the garbage collectors of the immune system, detect the FPZ portion of the IgG antibodies. The macrophages literally take bites out of the red cell membrane or engulf the cell entirely.
It's a microscopic slaughterhouse in the spleen.
It is. And as the macrophages digest the red blood cells, they break down the hemoglobin molecules. The hemoporphyrin ring is metabolically converted into a yellow pigment called unconjugated bilirubin. Normally, the liver processes this bilirubin and excretes it into the bile. But in a hemolytic reaction, the sheer volume of destroyed red cells overwhelms the liver's conjugation capacity. The excess yellow bilirubin spills out into the blood and deposits in the fatty tissues of the skin and eyes, causing the visible jaundice.
It's incredible how the visual symptom is just a byproduct of an overwhelmed waste disposal system. Let's look at a reaction that attacks the lungs, TRALI, transfusion-related acute lung injury.
TRALI is arguably the most feared complication today because it is one of the leading causes of transfusion-related mortality. The onset is rapid. Within 6 hours of the transfusion, the patient develops severe hypoxemia. They suddenly cannot oxygenate their blood. Their chest x-ray shows massive bilateral infiltrates. Their lungs are essentially filling with fluid.
And the mechanism behind TRALI is a complete inversion of everything we've talked about so far. Usually, it's the patient's immune system attacking the donor cells. Here, the donor blood is the aggressor.
Yes. It is a terrifying Trojan horse scenario. TRALI is predominantly caused by antibodies present in the donor's plasma that attack the recipient's immune cells, specifically HLA antibodies or human neutrophil alone antibodies in the donor plasma.
How does a blood donor get these antibodies in the first place?
They're typically formed during pregnancy. When a mother is pregnant, she is exposed to the paternal HLA antigens present on the fetal cells. Her immune system builds antibodies against those paternal antigens. If she later donates plasma, those potent HLA antibodies are sitting right there in the bag.
So we hang the bag, the donor plasma enters the patient. What happens when those antibodies reach the lungs?
TLI is often described as a two-hit mechanism. Hit one is the patient's underlying illness, maybe they have an infection or underwent surgery. The stress primes the patient's neutrophils, a type of white blood cell, causing them to sequester and gather in the tiny pulmonary capillaries of the lungs.
Okay.
Hit two is the transfusion. The donor's HLA antibodies circulate through the bloodstream, reach the lungs, and violently bind to the patient's primed neutrophils.
They activate the neutrophils inside the lungs.
They hyperactivate them. The neutrophils degranulate, releasing toxic enzymes and reactive oxygen species directly onto the delicate endothelial lining of the pulmonary capillaries. They burn holes in the capillary walls. Fluid from the blood rapidly leaks out into the alveolar air spaces. The patient essentially drowns from the inside out.
That is horrific. If donor plasma is the weapon, shouldn't we just screen every single donor for HLA antibodies?
If we did that, we would decimate the blood supply because millions of women have these antibodies and are perfectly healthy. Instead, blood centers instituted a massive systemic mitigation strategy. We now predominantly manufacture plasma and platelet products, which contain high volumes of plasma exclusively from male donors or from female donors who have never been pregnant.
Oh, that makes sense.
By restricting the plasma pool to populations highly unlikely to have HLA antibodies, the incidence of TROI has plummeted.
That is a brilliant systemic fix. The last reaction we need to cover is TACO, transfusion-associated circulatory overload. The symptoms are tachycardia, a racing heart, and spiking blood pressure.
This reaction is unique because it isn't immunological at all. There are no antibodies, no complement cascades, no macrophages. It is a purely mechanical problem of physics and plumbing.
Too much fluid, too fast.
Exactly. When you transfuse a unit of blood, you are rapidly expanding the volume of fluid inside the closed loop of the cardiovascular system. In a young, healthy trauma patient, the heart easily stretches and accommodates the extra volume.
But not for everyone.
Right. But in an elderly patient with congestive heart failure, or a small infant, or a patient with severe chronic anemia whose body is adjusted to a low blood volume, the heart muscle is stiff or weak.
It can't pump the extra fluid forward. It can't. The hydrostatic pressure backs up, the heart races to try and keep up, blood pressure spikes, and eventually the pressure forces fluid out of the pulmonary veins and into the lungs. It looks clinically very similar to TRAIL-AI. The patient is gasping for air, but the cause is completely different.
So what's the treatment?
The treatment isn't to wash the cells or use antihistamines. It is to immediately stop the transfusion, sit the patient upright, and administer potent intravenous diuretics to force the kidneys to excrete the excess fluid as urine.
It is a humbling reminder that sometimes, amidst all the high-level biochemistry, you just have to pay attention to basic hydraulics. And this brings us to our final frontier. Because these massive antibody battles, these complex compatibility matrices, they don't just happen during a transfusion in an adult hospital bed. They happen naturally across the placental barrier between a mother and her unborn child. This is the domain of hemolytic disease of the fetus and newborn, or HDFN. It is the intersection where immunohematology becomes a critical part of obstetrics. HDFN occurs when a maternal antibody actively crosses the placenta, enters the fetal circulation, and systematically destroys the red blood cells of the developing fetus.
But not all antibodies can cross the placenta, right? This goes back to the architectural differences we talked about in the antibody zoo.
Precisely. The placenta is an incredibly selective filter, specifically the syncytial trophoblast layer. It actively transports certain molecules to give the baby passive immunity. IgM antibodies, like the massive pentamer structures of naturally occurring anti-A and anti-B, are physically far too large to pass through the placental receptors. They are trapped in the maternal circulation.
Okay, but IgG?
But IgG antibodies, those tiny monomeric tweezers, are actively grabbed by specialized receptors on the placenta and shuttled across into the fetal bloodstream.
So if a mother has formed an IgG alone antibody directed against a blood group antigen that the baby inherited from the father, that antibody will be actively ferried across the barrier where it will hunt down the baby's blood cells.
Yes. And we generally categorize HDFN into two main physiological profiles, ABO-HDFN and RH-HDFN.
Let's start with ABO. It happens when a Group O mother has a Group A or Group B baby. But wait, earlier you said anti-A and anti-B are IgM grappling hooks. Why are they crossing the placenta?
This is a fascinating immunological quirk unique to Group O individuals. Group A and Group B individuals do produce large IgM antibodies, but the immune system of a Group O person who lacks both A and B antigens produces anti-A, anti-B, and a unique cross-reactive antibody called anti-AB.
Oh.
And for reasons we still don't entirely understand, this combined anti-AB antibody is predominantly the small IgG type.
Which means the Group O mother's anti-AB can fit through the placental receptors and reach her Group A or B fetus. Does this destroy the baby's blood?
It does attack the fetal red cells, but AVO-HDFN is almost always very mild. Often the only symptom is that the newborn becomes slightly jaundiced a day or two after birth and might need to spend some time under the blue phototherapy lights to help their immature liver break down the bilirubin.
Why is it so mild if the antibody can cross over?
Two brilliant biological buffering mechanisms. First, the A and B antigens on fetal red blood cells are not fully developed. The carbohydrate chains are sparse, so there aren't many targets for the antibody to hit. Second, and most importantly, A and B antigens are not restricted to red blood cells. They are ubiquitous tissue antigens found on endothelial cells, epithelial cells, and in tissue fluids all over the fetal body.
So the maternal antibodies cross the placenta, but they get completely distracted. They bind to the baby's tissue cells instead of concentrating all their firepower on the red blood cells.
Exactly. The tissues act as a sponge, absorbing the brunt of the maternal antibody attack. But Rh-HDFN, specifically caused by maternal anti-D, is a drastically different, much more lethal story.
Because the D antigen doesn't have those tissue sponges to protect it.
Correct. The D antigen is expressed exclusively on the membrane of the red blood cell. Furthermore, it is fully developed and densely packed on fetal cells very early in the first trimester. There is no distraction. Every single molecule of maternal IgG anti-D that crosses the placenta acts like a laser-guided missile, locking directly onto the fetal red blood cells.
And the fetal spleen starts chewing up those coated red cells.
The destruction is relentless. The fetus becomes profoundly anemic. To compensate, the fetal liver and spleen enlarge massively as they desperately try to manufacture new red blood cells to replace the ones being destroyed. Eventually, the fetal heart fails from pumping thin, oxygen-starved blood, leading to massive fluid accumulation in the fetal tissues, a condition known as hydrops fetalis, which is often fatal.
It is a devastating cascade, but it is entirely preventable now. This is where one of the greatest medical triumphs of the 20th century comes in. RhIg, better known as RhoGAM.
The development of RhoGAM fundamentally altered the landscape of maternal-fetal medicine. The goal of RhoGAM is to passively prevent an Rh-negative mother from ever forming her own permanent immune memory against the D antigen.
Let's walk through the mechanics of how it actually prevents that memory. An Rh-negative mother is carrying an Rh-positive baby. During a normal pregnancy, the blood systems are mostly separate. But during delivery,
as the placenta separates from the uterine wall, a feto-maternal hemorrhage almost always occurs. A small volume of the baby's Rh-positive blood spills over into the mother's circulation.
Okay.
The mother's immune system detects this foreign D antigen and begins the slow process of building a permanent army of anti-D memory B cells. The first baby is already born and safe. But any subsequent Rh-positive pregnancy will be relentlessly attacked by that permanent army.
So giving RhoGAM is like sending in a specialized decoy squad to sweep up the baby's rogue blood cells before the mother's immune system can even sound the alarm?
That is precisely how it works. RhoGAM is not a vaccine. It is a vial of highly concentrated manufactured IgG anti-D. We inject this manufactured antibody directly into the mother's muscle within 72 hours of delivery.
Wait, we are injecting the exact antibody we are afraid of.
We are, but we are injecting it passively. This manufactured anti-D floods the mother's bloodstream, rapidly locates the baby's Rh-positive cells, coats them, and signals the mother's macrophages to quietly clear them away.
Wow.
Because the cells are swept up and destroyed by the decoy squad, the mother's own B cells never get the opportunity to formally recognize the D antigen. She never forms permanent memory lymphocytes. A few months later, the injected RhoGAM degrades and vanishes, leaving her immune system as pristine and naive as it was before the pregnancy.
But there are absolute rules for when this decoy squad is useless. If the baby is born and we type the cord blood and the baby is Rh negative, we don't give RhoGAM.
Logic dictates that. If the baby is Rh negative, they lack the D antigen. The blood that spills into the mother has no D target on it. The mother's immune system has nothing to react to, so there's nothing for the decoy squad to sweep up.
And what if a mother comes into the clinic for her very first prenatal visit? We run an antibody screen, and we discover she already has a soaring titer of active anti-D. Can we give her RhoGAM to fix it?
Sadly, no. If she already has an active high-titer anti-D, meaning she was sensitized by a previous unrecognized miscarriage or a mismatched blood transfusion years ago, the permanent army has already been built. The memory cells are entrenched.
Too late.
Right. Giving her an injection of manufactured anti-D is like throwing a cup of water into a flooded basement. It does absolutely nothing to stop her own immune system from attacking the current fetus.
Okay, so if the mother is a candidate, she is Rh negative, her baby is R positive, and she doesn't have an active antibody, we know we need to give her RhoGAM. But how do we know how much to give her? If the baby had a massive hemorrhage during a traumatic delivery, 1 vial of the decoy squad might not be enough.
This is a critical laboratory calculation. 1 standard vial of RhoGAM provides enough passive antibody to sweep up exactly 30 milliliters of fetal whole blood. For a normal, uncomplicated delivery, the feto-maternal hemorrhage is usually less than 10 milliliters, so 1 vial is perfectly sufficient. But to be absolutely certain, we have to quantify the bleed.
We do a screening test first, the fetal rosette test.
Yes, the rosette test is a qualitative screen. It just tells us yes or no if a large bleed occurred. We take a sample of the mother's blood drawn after delivery. We incubate it with an anti-D reagent, which binds to any Rh-positive fetal cells floating around. Then we add specially treated indicator red cells. If a fetal cell is present, the indicator cells will cluster all around it, physically forming a microscopic flower or a rosette.
It's like dropping a biochemical dye marker on the rogue cells. You look under the microscope, and if you see a rosette, you know fetal cells are present.
Exactly. If the rosette test is negative, we issue 1 vial of RhoGAM and the mother goes home. But if it is positive, we know a bleed greater than 30 milliliters has occurred. We just don't know if it's 40 milliliters or 100 milliliters. To quantify the exact volume, we must perform the Kleihauer-Betke test.
The Kleihauer-Betke. This test relies on acid elution. Walk me through the biochemistry here, because it sounds like we are actively destroying the mother's blood on a slide.
We are doing exactly that. The Kleihauer-Betke test takes advantage of a fundamental structural difference between adult hemoglobin and fetal hemoglobin. Adult hemoglobin, known as hemoglobin A, is composed of 2 alpha and 2 beta protein chains. Fetal hemoglobin, hemoglobin F, has 2 alpha and 2 gamma chains. That gamma chain makes hemoglobin F incredibly robust and resistant to acidic environments.
So you smear the mother's blood on a glass slide, let it dry, and then what?
We submerge the entire slide into a citric acid phosphate buffer at a very specific pH of 3.3. At this highly acidic pH, the beta chains in the adult hemoglobin A rapidly denature. The adult hemoglobin physically dissolves and washes out of the cell membrane, leaving behind what we call a ghost cell, just the faint, empty outline of the red cell lipid bilayer.
But the fetal cells, with their gamma chains, resist the acid.
They hold completely firm. After the acid bath, we stain the slide with a red dye called erythrocin. The adult ghost cells, having no hemoglobin left to bind the dye, remain nearly invisible. But the fetal cells, still packed with robust hemoglobin F, absorb the dye intensely. When you look under the microscope, the fetal cells light up as vibrant, bright pink circles standing out against a vast, pale sea of maternal ghost cells.
So the technologist literally sits at the microscope and counts bright pink dots to figure out how much the baby bled.
Yes. It is painstaking work. We count exactly 2,000 cells on the slide. We count the number of pink fetal cells and divide it by the 2,000 total cells to get a percentage. Let's say we find 20 fetal cells out of 2,000, that's 1%. We then plug that percentage into a clinical math formula.
How does the math work to get the number of vials?
We assume the average maternal blood volume is 5,000 milliliters. We multiply 5,000 by our 1% to determine that 50 milliliters of fetal blood are circulating in the mother. Remember, 1 vial of RhoGAM covers 30 milliliters. So we divide 50 by 30, which gives us 1.66 vials.
You can't give 0.66 of a vial, so you round up.
The clinical rule is highly specific to ensure a safety margin. If the decimal is 0.5 or higher, you round up and add 1 entire vial for safety. So 1.66 rounds up to 2. We add 1 for safety. The mother received 3 vials of RhoGAM. If the math had yielded 1.2 vials, we would round down to 1, add 1 for safety, and issue 2 vials. It guarantees there's an overwhelming force of decoy squad antibodies to sweep up every last rogue fetal cell.
That is an incredible intersection of biochemistry and clinical mathematics. There is one final piece to this HDFN puzzle. When a baby is born, the lab tests the blood taken from the umbilical cord. You test for ABO, you test for Rh, and you do a date to see if maternal antibodies are coating the baby's cells. But you never, ever perform an antibody screen on the cord blood plasma. Why?
Because a newborn infant is immunologically naive. A fetus does not possess the cellular machinery to manufacture their own robust allo antibodies. They do not start producing their own significant immunoglobulins until they are about 3 to 6 months old.
So if you take a newborn's plasma, run an antibody screen, and it comes up glaringly positive, whose antibody are you actually looking at?
You are looking at the mother's antibody. You are just measuring the maternal IgG that actively crossed the placenta. Performing an antibody screen on a newborn is a complete waste of time, reagents, and healthcare resources because it only tells you what is circulating in the mother. And we already comprehensively tested the mother when she was admitted to the labor unit. We only perform the dates on the cord blood because that tells us if the maternal antibody has physically crossed over and attached to the baby's red cells, causing active HDFN.
Okay, let's step back and look at the whole picture. We have gone on an absolutely massive journey today. We started by dismantling the simple idea of blood types, looking at the FUT1 genetic blueprints, the concrete slabs of the H antigen, and the missing docking ports of the Bombay phenotype. We navigated the Fisher race and Wiener genetics of the Rh system, and the structural puzzle pieces of the D mosaic.
We ventured into the antibody zoo, comparing the massive IgM grappling hooks with the tiny IgG tweezers. We explored the physics of the Goey-Chapman layer, using LISS to dehydrate the environment and bridge the zeta potential.
We tracked the kid ninjas hiding in the lymph nodes, ready to launch an anamnestic response, and the biphasic Doneth-Landsteiner hemolysin trapping red cells in the cold to blow them up in the warm. We saw how the polyspecific AHG footprint proves a complement attack occurred, and how, tragically, the deadliest thing in a multi-million-dollar blood bank isn't a rare antibody, but a misspelled name on a tube.
And finally, we saw the breathtaking mechanics of the placenta, the acid resistance of fetal hemoglobin, and the medical marvel of ROJAM acting as a passive decoy squad to protect generations of children from hemolytic disease.
It is a staggering field of science, and I want to leave you with one final thought to mull over. Think about that zeta potential, the literal cloud of electrical charge surrounding every single one of your billions of red blood cells. Right now, as you listen to this, your heart is pumping those cells through thousands of miles of microscopic capillaries. They are rushing through your body, bumping into each other, but that invisible electrical force field is violently bouncing them apart, preventing you from clotting into a solid block of tissue.
It is the physics of life itself, happening millions of times a second.
Blood banking isn't just mixing red liquids in a tube. It is the high-stakes manipulation of microscopic electrical force fields, genetic memory, and evolutionary defense systems. Next time you get a paper cut and see a drop of blood, remember the universe of microscopic combat and architecture happening inside it. Thank you for taking this deep dive with us today. Keep exploring the science of yourself.